welcome to everybody. This is the third Thrombosis and Thrombocytopenia syndrome patient and public involvement and engagement session. And I should explain, I am Professor Beverley, founder and trustee of the meeting with the Research Consortium. The whole point of the meeting is to allow those who've been diagnosed with vaccine-induced immune thrombocytopenia and thrombosis, or people looking after patients or individuals with this problem, or sadly those who have lost a loved one as a result of it, to hear about the research that's been going on. We've got a number of speakers, and Pip is going to tell us a little bit more about the consortium in a minute. My role, well, I was very much involved in the early days of VITT, and with 4 other haematologists, we formed the expert haematology group, and we offered help with every case of VITT by setting up a daily call from March 2021 for all the doctors in the UK who could sign in, and we could discuss each case individually and offer best care. The rules of this meeting are we will be filming all the presentations so that they can go up on the Thrombosis UK website later, but we will not be filming between, and this is an opportunity for people to ask questions confidentially, and we will not be discussing the questions afterwards. So it's a real opportunity for those listening. So I'm going to hand over now to my co-chair, Dr. Pip Nicolson, to introduce himself and to tell you a little bit more about the research. Pip, over to you.
Thank you, Beverley, and thank you all for those who are speaking, and particularly those who are participating and listening. And we hope that you are able to learn something or find things that are useful that come out of this meeting. My name's Pip Nicolson. I'm a haematologist as well, but from Birmingham. And I've been a relatively junior member of the consortium that was set up to perform research into vaccine-induced thrombosis and thrombocytopenia, VITT. To clarify, to give you a bit of background about the grant, it was awarded by the National Institute for Health Research, which is a sort of government organisation to promote clinical research in the UK. And it was funded to a big group of different institutions across the UK, so including Birmingham, Bristol, Cambridge, Cardiff, Edinburgh, Liverpool, and universities in London. But as well as universities, it was also granted to other public institutions such as Genomics England and Health Data Research UK and Public Health England. So it was a big collaborative group between all these and people within all these institutions. Importantly, it didn't involve anybody from Oxford University because of conflicts of interest there. But it was set up in order to investigate primarily the underlying reasons for why these clots and low platelets and causing this devastating condition, why they happened. And it was separated into 5 work packages. And we have a representative from each of the work packages here to give a short lay summary of the work that has been done since that grant was awarded, because the grant's now come to completion. So the work from that grant is coming to an end, and sort of what have they, that's why, so they've got an opportunity to present to you to say what have they found and how is this going to help going forward. So that's the background to the meeting. I'm going to then hand back to Beverley, who's going to start by introducing our first speaker to you.
Thank you, Pip. So our first speaker, Lara Edwards, is Programme Director for National Core Study Data and Connectivity at Health Data Research UK. And the aim of her work is to address the awful fragmentation of health data by working with all the major institutions across the 4 nations to organise medical, biological and social science data on a UK-wide scale. So thank you very much, Laura, for coming this afternoon.
Thank you, Beverley, and good afternoon to everyone, and thank you very much for inviting me along to speak. So thank you very much for Beverley's introduction as well. While I'm sharing my screen, I'll just say a couple of words about HDI UK. So as Beverley very clearly articulated, our kind of mission and aim is to harness the power and harness the power of national and regional level linked data to improve health and care outcomes. And at the moment, it's a very fragmented picture across the UK, but we're really trying our best to try to change that. And I'll explain a bit more about that as we go through the presentation. So the purpose of our work really was twofold, really. So one, there was work led by Professor Cathy Sudlow and Dr. Will Whiteley and others through the British Heart Foundation Data Science Centre, which is part of HDI UK. And they were really looking at and trying to identify background rates and incidents of VITT occurring across the UK in national level data. And using the kind of national level, and I'll go into a little bit more explanations to what I mean by national and regional level data. They really wanted to evaluate the association between COVID-19 infection and thrombotic and blood clotting disorders, and also establish these kind of the background rates and association of these disorders with COVID-19 vaccines. And as I'll explain more clearly in a moment, there are limitations really as to what we can do with national level data at the moment when we're trying to identify events or adverse events or diseases or any kind of public health and emergency event in near real time. Because national level data at the moment is available to researchers with a bit of a delay. So at the same time, as we were analysing the national level data, we really wanted to explore what is available at regional level data and how we could harness that more effectively across the country to provide kind of more detailed information and provide data to researchers more rapidly. So all of our research is data-led, so we are not participating, we're not recruiting participants in clinics, or we're not doing any lab research. But the purpose of our research is to bring together hospital data, vaccination data, primary care data, for example, to link that and really get a picture of what's going on. So what do I mean by health data? That's a bit more complicated than it first seems. So at the moment, obviously, when any of us attend a hospital appointment or go and see our GP, we know that there are electronic health records where our data is entered, whether that's in a hospital or a GP. And there may be data that's entered via imaging or laboratory data, etc. as well. And all to be useful for research, all of this data has to come together and be coded. So, for example, for diabetes, there'll be a specific code for specific types of diabetes, and similarly for any other diagnosis or prescription or intervention. And that's really key for researchers to be able to use data in a useful way. And at the moment, all of our kind of healthcare data from hospital systems and GP systems is forwarded to our national data custodians. So in England, that's NHS England, and Scotland, that's NHS Scotland, and equivalent across Wales and Northern Ireland. So at the moment, that's where when researchers want to access national level data, they access it via these national data custodians who look after our data for us. And in addition to that, there are regional research databases. So the example of one I've given down here is something called DataLock, which is in Edinburgh and NHS Lothian, and they tend to hold really detailed and granular data for their own population. And finally, there's something called research databases. So the example I've given down there is the Scottish National Data Safe Haven, and there's lots of GP primary care research databases as well that researchers can access. So it's quite a complex and fragmented picture. And just a quick note as to how researchers access data securely and how your data is kept safe. So whether researchers are accessing data at a national level, so they can access national data through NHS England via a secure data safe haven or a trusted research environment, or they can access data at the regional level. So for example, for London, for Birmingham, or for regions in Scotland or Wales or Northern Ireland. They can access kind of data safe havens at that regional level as well. And TREs, or trusted research environments, really just make our research safer. The key thing is that the data never leaves that environment, and researchers are assessed to check that they are approved and they're trained to access data, and the projects have got ethical approval with really clear potential for a clear public benefit. So they really provide researchers with this kind of single location to access valuable data sets and ensure that the data and analytical tools are all in one place, a bit like a secure reference library. So I'm just going to summarize really quickly on the work we did using the national level data sets. And really the advantage of using national level data sets are the numbers, so the population and the scale. So by using the NHS England Trusted Research Environment, or by using the Welsh Trusted Research Environment, collectively, we were able to analyse data in more than 50 million people. So you can see immediately the scale and the value of that. And as we all know, at the start of the COVID-19 pandemic, there was research from the outset that suggested that coronavirus may lead to blood clots and strokes in lots of different outlets and newspapers. And as the kind of pandemic developed, that kind of the reasoning and why coronavirus was causing these blood clots in young and and healthy people was a bit of a mystery. So for the first part of our research, if you remember, was looking at the risk of clots with COVID-19, and the second part was looking at the association of clots associated with COVID-19 vaccines. So this is the data we looked at. We looked at data for everyone registered with a GP in England and Wales from January 2020. We looked at all diagnoses of COVID-19, whether that be in the community or in a GP or a hospital. And then we looked at all diagnoses of blood clots through hospital records and GP records. So that was clots in veins of lungs and legs, or clots in arteries of the brain and the heart. And what did we find? So we found that 1 to 2 weeks after COVID-19 infection, there was a 20-fold risk of heart attack or stroke. And that decreased as time went on, moving from 1 month to over a year to 4 times and 1 times. And then we looked at the increased risk of clots in legs of the lungs after COVID-19 compared with no COVID-19. And you can see down here across in blue, there's quite a significant increase in the 1 to 2 weeks time period since infection, and that decreases as time goes on. And at the beginning of the COVID-19 vaccine programme, quite quickly there was identification and alerts came out, concluding that there may be a possible link between the AstraZeneca vaccine in particular and blood clots. And that quite quickly made its way into the news and into academic journals. And this journal was with the work of Beverley Hunt and our colleagues, who had first identified VITT in the UK within this clinical hematologist expert group, and reported on the first cases. So how risky were COVID-19 vaccines is what we wanted to ask, again using this national level data. So again, we looked at everyone registered with GP in England in January 2020. We looked at all COVID-19 vaccination data. and we looked at all diagnoses, patients who did a vaccine, and any diagnosis of rare blood clots and low platelet counts. Low platelets are really important for blood clotting. So what did we find then? So we looked at both AstraZeneca and we looked at Pfizer vaccines using the national data. And we found that the AstraZeneca vaccine increased the risk. This is what this table is telling us here. It increased the risks of clots in the brain by a small amount. And you can see the risk going down as time goes on, but the risk increasing as we're increasing up to 28 days. We also found that it increased the risk of low platelet levels. And as I just said, platelet levels are really important for blood clotting. And we found that it increased the risk of low platelet levels by a small amount. We also looked at the risk of common health conditions, so clots in legs or lungs, and heart attack or stroke. And we noted that they were around 20 to 40% lower in people over 70 and about 25% lower in people over 70. And overall, our conclusions using the national data was that COVID led to a higher risk of strokes soon after infection, and AstraZeneca vaccination led to a small increase in the number of rare clots in younger people. But overall, the clots were reduced. However, with national data comes limitations. And I'd mentioned that at the very outset, and this is why we really wanted to use to explore the capability of regional level data. VITT is not coded very well or described very well in national level data sets. So it was quite hard for researchers at some point to really get well-coded data for specific VITT diagnoses. In addition, there is this time lag of data being available. So data available now from NHS England, from national trusted research environments, are often only available to researchers after a patient has left hospital, and with a 6-week time lag delay. And when researchers are trying to identify something like that, or trying to identify something that's happening in the population very rapidly, it's obviously really helpful to get access to data as soon as patients are turning up to the emergency department. And this is more possible with regional linked data, but as we found, the picture is very fragmented. And there are other data feeds that would be really helpful when looking at something like VITT that aren't available at a national level. So, for example, laboratory data. So what we concluded really was that a really rapid, near real-time hospital system data feed would really provide vital data for programs such as this, when we're really trying to identify evaluation, safety, and effectiveness of medicines, and identify adverse events that are happening in the population. So what did we do then to do this? So we convened a collaborative group of 9 regions across England and Scotland. And we picked these regions because we know that each of them have really good regional and secure data safe havens or trusted research environments already. They're all at different kind of levels of how digitally mature they are. But we wanted to have a really deep delve into these and map the data capability and availability in detail, and really map variances and other aspects. It's really important for research, such as how researchers access that data securely. And we wanted to get feedback from the public. How do they feel about the use of their regional data being used for approved research? We had a workshop with members of the public, and that was UK-wide to get feedback, really. So we wanted to understand how people felt about their how researchers accessing their data, their regional data that's held regionally by, for example, the general practitioner, and how they felt about that being used for research and used by researchers outside of that region. And there was really strong support for that. And actually quite a lot of members of the public were surprised that we weren't doing this in a more joined up and coherent way already. Obviously, one of the key things that came through was that it has to be absolutely clear what the benefits to the public, NHS and researchers are, and ensuring that who is accessing data and why and for what purpose was really secure and transparent. And another key theme that came up from researchers was that actually they really wanted, if if the regional level data was really helpful and it was more detailed and it was available in more real time, they really wanted researchers to access it. So what have we found then? So we found that there's a lot of variation across regions on what types of data are available, how it is coded, and how rapidly it is available. And for the VITT case, what we did was implement a computer algorithm across each of those different regions, just to see how quickly we could identify known VITT cases in those hospitals. And that was really effective. But what was really difficult was still this huge variation. So, for example, some of the regions had really well coded and available laboratory data, and others didn't. And some of those data sets were really needed to drive forward to identify VITT cases very rapidly. So we found also that there is real variances in how securely and how researchers can actually access this data across multiple regions as well, and how rapidly it is available. I spoke earlier about the need for near real-time data in an event such as VITT, and at the moment that was quite still mixed across the UK. So further developments, the good news is that NHS England have just funded a lot of funding into really developing this regional level capability. So we hope within the next year or so there'll be real differences in data capability, how different regions can interoperate with each other, how researchers can access data across multiple regions, and ensuring that researchers have got really streamlined data access processes to be able to do this. So I think that's it for me, and I'm happy to take any questions or comments.
Thank you very much, Lara. Thank you for sort of explaining how complex, making a complex topic understandable. Thank you. I'm going to introduce our next speaker now, who is Dr. Stephen Winchester. He is a consultant virologist at Frimley Park Hospital in Surrey, and he sits on the UK HSA expert panel on COVID-19. He is going to talk to us on understanding the significance of the clinical testing positive for these anti-PF4 antibodies, both within VITT, but also outside of the context of VITT. So, Stephen, do start.
Thank you. I'm just sharing my screen. Thanks for the introduction. So yes, I'm going to sort of go back to the start of this project in terms of the background and what we intended to do. What was known, you mentioned PF4, what was known about platelet factor 4, that it's a small cytokine, so involved in signaling, and it's released from alpha granules of activated platelets during platelet aggregation, as has been touched on regarding platelets and their involvement in clotting. And so it then promotes clotting by moderating the effects of heparin-like molecules. And so the reason for this study was that the development of anti-PF4, antibodies to PF4, were considered to be a defining feature of a newly described thrombosis and thrombocytopenia syndrome that had been reported after receipt of the AstraZeneca COVID-19 vaccine or the Janssen COVID-19 vaccine. And a high level of these PF4 antibodies were detected using an ELISA, so a test that uses an enzyme reaction in order to detect the antibody, and then the enzyme reaction then produces a color change in a positive result, for example. And this was described in the majority of cases in Germany, Norway, and the UK. And so the underlying assumption was that perhaps administration of the vaccine led to development of platelet activating antibodies, and that platelet activation then led to prothrombotic disorder that had been described. However, the low rate of the observed event suggests that specific host or other cofactors are required to either develop these antibodies or to develop any pathology. So the purpose of this study was to measure the background rate of PF4 antibody in healthy unvaccinated individuals, also to measure this in vaccinated individuals and following COVID-19 infection as well, and to compare the usefulness of the different assays and tests for antibodies to PF4. So at the time, the commercial assays were available, but though not all of these were known to detect the TTS-associated PF4 antibody. Immunocore assay anti-PF4 was highly sensitive and so used for the study. The background population prevalence of antibodies to PF4, though, is uncertain, and rates have been reported between 1 and 6.6%, dependent upon not only the tests used, the populations tested as well, and what cutoff is determined, what is defining positivity. So UKHSA collects samples for seroprevalence studies for COVID-19 and other vaccine preventable diseases through its sero-epidemiology unit, including weekly testing of blood donor samples, samples from primary care as part of routine blood testing, residual sera from participating NHS laboratories, and specific vaccine studies. So all these different sources of obtaining samples to be used in the study. And so essentially to measure the background rate of PF4 antibody in healthy unvaccinated individuals, around 2,000 samples were obtained from COVID seronegative individuals, so those who had not been exposed to COVID, and then around 2,000 from those who had blood from blood donor samples. To measure the prevalence of high levels of antibodies to PF4 in vaccinated individuals, and around 2,000 samples from adults linked to vaccine type and date. So this was determined whether there were high levels of these. And then, as I mentioned as well, the study was also to compare the usefulness of different assays for antibody to PF4 detection. So around 200 samples. And so this is the distribution of the pre-pandemic samples, and there are two cutoffs, the lower cutoff being the cutoff of the assay, and then another level set higher up that was thought to be of greater significance at this level. And so this was before pre-pandemic, and then these were similarly from those that had confirmed COVID-19 infection, and then subsequently the other samples following vaccination. And essentially there was no evidence to suggest that the vaccine commonly provokes production of these antibodies by comparing these as an overall description. For the purposes of the study, 3 assays were selected, and one of these was currently unavailable due to a material shortage, so 2 assays were used. And all of the positive samples, both significance levels, were tested, and then also retesting in the original assay as well. I don't have a slide of the comparison of the different assays, as due to availability they were a bit delayed in being obtained, but it's all been tested now and just on the final analysis. So that concludes my talk. So thank you to everyone at UKHSA for all of their work with this. Thank you.
We'll move on to the third of the 5 work packages from the consortium. So this talk is going to be given by Simon Abrams. He is a senior lecturer at the University of Liverpool and has his research focus is on bleeding and clotting, particularly in bleeding and clotting that's associated with patients who are severely ill in the intensive care unit, of which many patients with VITT were. And he also has expertise in understanding how the blood, in the white cells, the neutrophils, are involved in causing clots. he's going to speak to us about the relationship between the immune system and clotting that happens after COVID vaccines. So Simon, the floor is yours.
Thank you. And can everyone see the slide nicely? Good.
We can.
So, hello from Liverpool, the host city for the Eurovision Song Contest. I'll be heading down there later on today to enjoy the festivities. But the focus now really is to present our findings to you. And really, what I want to be talking about today is the mechanism of enhanced clotting in VITT patients. Okay, so when clotting normally occurs, you get cellular activation and damage that's very local to where the actual injury has occurred. And this causes the leakage of proteins that come out of the damaged area that can initiate the clot formation. However, what we found is that if you get a lot of cellular death, so like the whole body's injured in a way, then proteins can leak out of these damaged cells and cause coagulation or clotting to happen throughout the body. So really what we wanted to do here in Liverpool was to see whether that actually could be the case in VITT patients. So the first indicator that this could actually be happening is we saw something called lymphopenia. Now what this is really you get a reduction in your white blood cells within the circulation. And a lot of the time this can be attributed to the cells actually dying. So tick, that's one. Perhaps that the cells are dying because of those evidence. However, a more compelling one was actually that we saw a marker called LDH. Now, what this is, so if you see on the left-hand side, this is sort of like your cells dying and all sorts of things are getting released out of the cells that are damaged. And LDH is one of them. It's a marker that's used clinically as cellular death. And what we can see in the VITT patients compared to AstraZeneca pre and post vaccine controls, we're seeing damage marker elevated in the circulation of VITT patients. Now, why am I talking about cell death? Well, here in Liverpool, we look into a protein or a group of proteins called histones. Now, what are histones? Well, they're typically found within your cell nucleus, so the brain of your cell. Now, what their role is within the cell nucleus is the DNA is wound around these histones that cause it to be very tightly compacted within the cell nucleus. However, when cells die, these histones can be released into the circulation. Now, why is this important? Because we found that when there's a lot of cellular death, there's extremely elevated histones within the circulation, and these proteins can be toxic. When I say they're toxic, well, what they can do is actually damage the cells that line your blood vessel walls. They can actually cause platelet counts to drop. Now, this is something that we see in the VITT patients. We see also that histones can directly induce clot formation themselves, as well as initiating the clots. They can also cause the clot formation to be disseminated throughout the entire body. Histones are pro-inflammatory, and in fact, they can also induce organ injury. And importantly, in the ICU or the intensive care units, we find that elevated histones are associated with increased mortality. So what about circulating histones in VITT? Well, it's quite apparent here from the top left-hand panel that circulating histones are extremely elevated in patients with VITT compared to the control patients. What we find is that histones were really associated, sadly, with poor outcome in VITT. And if we were measured circulating histones on the admission to a hospital when the VITT patients were unwell, we found that by measuring circulating histones, we were able to independently predict mortality in VITT, irrespective of age and gender. Now, if you look at this final panel at the bottom right-hand side, we used a predetermined cutoff for histone levels within the circulation that we've characterized in other critically ill diseases. We find that those patients with relatively low levels of histones, the majority of them survived. And those patients with extremely high levels of histones, sadly, these patients have passed away. Now, I'm going to talk to you briefly now about a new mechanism that's part of the host response. And not only does it give you beautiful pictures, but also it's really interesting, the science behind it. So this image on the top left-hand side here is something called a neutrophil extracellular trap. Now, what is this? Well, it's when a neutrophil gets stimulated by bacteria or some sort of organism that's infecting your body. Now, the role of these neutrophils is to trap and to kill any invading microorganism. Now, to do this, it releases its DNA from inside the cellular nucleus outside of the cell, and it can be decorated with enzymes that kill off bacteria, such as histones. So that's the reason that I bring up the nets at this point. Now, what we found actually is that by taking plasma or serum, so the liquid that's in your blood when you remove all of the cells and incubate it with neutrophils from healthy donors, we're able to see here that in fact there must be things in the circulation of VITT patients that can induce these nets within them. Well, why is this important? Because not only are nets important for trapping and killing bacteria, but also if you imagine you have these sort of web-like structures within your circulation, they can trap and kill your cells as well. So these can cause microvascular occlusion or clots within your organs. Now, unsurprisingly, in the literature, it was demonstrated that actually we could find nets in VITT patients' cerebral sinus vein thrombi, which is stained here by these multicolor images. Well, why are histones and nets important? Well, our group here has demonstrated that both circulating histones and also nets are really associated with the dissemination or the spreading of clots around your body, particularly in patients that are in the intensive care unit. So next, what we wanted to do is see whether, in fact, because VITT patients have elevated histones and they are able to, the samples can induce nets formation, do VITT patients actually score for disseminated intravascular coagulation? Now, the reason I say this is because VITT patients typically have a reduced platelet count, reduced fibrinogen levels, extremely elevated D-dimer. Well, D-dimer is a protein that is released into the circulation when your clots break down. So it's a marker of clot formation within your body. And what we find is, in fact, yes, patients with VITT not only have localized thrombi, but they really have a disseminated coagulation around their body. More evidence of this, in fact, is what we're able to see is that we can detect circulating microclots in VITT patients. And I must emphasize that this has been done in samples when people were immediately hospitalized, and we haven't done any further work into looking at whether there's any long-term microclots in VITT. Finally, I just wanted to show here that we can see the nets are deposed within the kidney of VITT patients. So not only can you see them local within the clots that have been described previously, but we can actually see nets that have been disseminated and deposited within vital organs. Not only this, but we can see the clots are actually present within these organs as well. So really what I want to hit home is that not only was VITT a sort of localized thrombosis, but what we've evidence from our work is really showing is that it was disseminated throughout the body, typified by low platelets. We see clots, we see cellular death, which results in histone release. And we think that these histones really are key players in causing these low platelets and disseminated clot formation. And just finally, I'd like to really say that hopefully within the future, by targeting histones, we may be able to prevent the sort of adverse outcomes associated with VITT. I'd like to thank you all for your attention and also my funders.
Thank you very much, Simon. Pleasure as always to hear you speak. Next, Dr. Richard Buka, who is a clinical research fellow in haematology. So he's a doctor and a researcher at the University of Birmingham. His main research interest is in the role of this platelet factor 4 in clotting. But he's going to talk to us about how that factors into how platelets are activated and then reduced in patients with VITT.
Good afternoon, everyone. Thanks for giving me the opportunity to talk and just share what we've been doing over the last couple of years. So we're predominantly a group that focuses on the biology of platelets. So we attack the problem of VITT and trying to understand how VITT occurs in the body by looking at platelets. On the slides. So just as a recap, and I'm sure many of you have looked into exactly how VITT and how these antibodies cause such problems in the body. After a vaccination with a adenoviral vector vaccine, and the vast majority of people with VITT in the UK will have received the AstraZeneca vaccine, we see this molecule PF4, so platelet factor 4, which is a very abundant protein that comes out of platelets. We see that that can glue onto the viral particles, the particles in the vaccine. Somehow, we don't exactly know how, somehow the body produces very strong antibodies against this molecule, such that it can join these platelet factor 4 molecules together into what we call immune complexes. Now, immune complexes are common, but in this situation, they are able to bind to platelets and to white blood cells and cause all sorts of problems. So as Simon said before, when they bind to white blood cells, they can cause netosis, so the release of lots of DNA and other cellular fragments into this bloodstream and cause inflammation and blood clotting. And this, we think, is a really important factor in the pathology of VITT. But as I said before, our group is a platelet group, so we're not interested in the white blood cells. They're not our friends. We look at platelets and we look at how platelets get activated. So after this process happens, we see white blood cells and platelets getting activated, which cause blood clots. Because those blood clots are very big and there's lots of them occurring, platelets get used up really rapidly in those blood clots. So platelets are a really important blood cell, essentially the plugs that go around our body. So if you cut yourself, the platelets are the first guys on the scene, stopping your blood seeping out and preventing bleeding. But what happens in VITT is you get blood clots, your platelets all get used up, and then because your platelets have all been used up, you then end up with bleeding, which is what we saw in many patients. But I just want to focus on this bit. So as with many things in the history of medicine, it's been small observations that have led to more interesting discoveries. So I'm going to focus in on exactly how these immune complexes activate platelets and cause clots. So early on in the story of VITT, Pip and his previous postdoctoral researcher in the lab, Chris Smith, worked on exactly how platelets were being activated, and they showed that this molecule, this immune complex, was activating this receptor called Fc gamma R2A. Now I expect you to all remember that, and we're going to quiz you at the end. But what we didn't anticipate is that we think that PF4 itself is activating platelets. Now, this is quite surprising. So Fc gamma R2A is already known to activate platelets in another condition called HIT, which has been discussed already and is very similar to VITT. So it was unsurprising that this is a mechanism in VITT. But what is much more surprising is that we found data, and we found that when we look at platelets in test tubes, using blood from patients with VITT, using antibodies from patients with VITT, we get activation through this receptor as well, this c-MIPL receptor. Again, another catchy name you'll be quizzed on. But I guess the question is, so what? And Pip's question before, how does this really help people? Well, the observation that PF4, which is a really, really abundant molecule in the body, is one of the key constituents of platelets. The observation that that molecule actually activates platelets is completely new to science. And without VITT, it's likely that it will be many, many years until we recognize that. Importantly, those pathways can be blocked. So the way that receptors work is that they stick out on the surface of blood cells, and then by various cascades, almost like a domino rally, it knocks over the first domino, and then all the others cascade downwards. And that's how you get cells to do things. And in platelets, when platelets get activated, they change shape, they become really sticky, and they release lots of chemicals that promote blood clotting. But we can block those downstream processes with drugs that already exist on the market. And the other thing is, we think this is relevant to other diseases. So particularly the PF4 story, the fact that platelet factor 4 activates platelets on its own, we think it's probably relevant to other diseases of blood clotting, and also how the body physically makes platelets. Because this receptor, c-MIPL, is also the receptor for the main hormone that drives the production of platelets in the bone. So that's what I've been working on. My colleague Sam Montague published this paper last year that looked at the follow-up of patients with VITT and looked at the antibody levels in patients with VITT. Now, again, as Pip said before, in HIT, which is this similar condition, we see antibody levels fall very quickly. And after around 100 days, the vast majority of patients don't have these antibodies detectable. But in VITT, you see anti-PF4 antibody levels being detectable at 7 months post. And we know from anecdotal things around the country, and from many of you will know that in patients with VITT, the antibodies remain detectable even now. And that makes this condition very different to HIT, although we often talk about them in the same breath. Again, why is that important? Well, it may guide decisions on whether to continue or to stop anticoagulation. But the problem is that there are very few patients with VITT around the country and around the world. Difficult to do that in a trial. But again, the importance of continuing to monitor all those patients with VITT and to publish the findings and to share that data. When people do stop their anticoagulation, and certain people continue, some people stop, being able to compare the difference and monitor the safety and monitor whether there are any problems ensuing from that is massively important. So thank you so much to all of you for partaking in research, for being interested in the research. And I know full well that although this was such a horrible condition and incredibly scary, I know that interesting science and very useful science is coming out of it. And I'd be more than happy to take any questions, either now or at the end. And that's it. Thanks.
Thank you, Richard.
Thank you. So yes, we're very lucky to have Sir Mark Caulfield. He's Vice Principal for Health and he's Professor of Clinical Pharmacology at Queen Mary University of London. He's President-elect of the British Pharmacological Society. He's made a major contribution to the discovery of genes related to blood pressure, cardiovascular disease, and rare diseases. And he was Chief Scientific Officer for Genomics England between 2013 and 2021. And he stepped down from this role to focus on leading Bart's Life Sciences, which is based in Whitechapel. So we're very lucky to have you, Mark, and very much looking forward to what you're going to tell us. Thank you.
Thank you so much for inviting me. It's been a privilege to work with all of you on this terrible disease and try and see what we can do to help patients together. So I'm just going to share just a few slides with you about what we're doing in the genomics element. And the fundamental question here is clotting associated with vaccination due to our genetic makeup? And so what we set out to do when we established this consortium under the auspices of the Medicines, Healthcare and Regulatory Products Agency, and the and Public Health England and NIHR, was to investigate that. Now, in COVID, we'd already been looking at severe illness. So asking the question, does your or my genetic makeup affect our susceptibility to critical illness caused by COVID-19? On the left at the bottom of your picture, you see the London Nightingale Hospital after the fit out was completed, and inside the Excel Centre there were 500 ventilated beds. Thankfully, we only ever used about 55 of them. And on the right, you see a vaccine trial centre, which we set up in a library to deliver vaccines. These were in trials of other vaccines, not the Oxford AstraZeneca, but this was regarded by, when we did public and patient engagement, to be a safe place that people would come, and they were too fearful to come to the hospital. So to answer the genetic question, and one of the reasons for showing you the Nightingale is that this required collaboration with multiple UK intensive care units. And we did that through the genomics study. And that looked at people who had been admitted to ITU with severe COVID. Mostly they had lung failure. Some of them had other organ failure. And one thing that they had was some of them had clots. And so the question really was, during the disease, was there something in us as humans inside your or my genetic code that might be making us susceptible to severe reactions? And could that be interfacing with the virus itself? And were there viral factors that could change that? So as a result of this proposition, which was in March 2020, the governments and other agencies that's listed here set aside some money to investigate this. And then when it became clear that there were also reactions to vaccination where unusual clotting and other features were being experienced by patients, we worked with our public and patient group, because we didn't have any with the ITT at that time, and set about framing a program with the other consortium members you're hearing from this afternoon. So this is the genomics limb. Now, why have I mentioned to you about severe COVID? Because in severe COVID, some people get clots. And one of the key questions we were investigating is there a susceptibility to clotting? And that is obviously something that could have relevance for vaccine-induced clotting. And that's one of the things we've been investigating. So to summarize what we found in severe COVID, we've now found in multiple studies in the UK, mostly in the UK, but also in the rest of the world, that there are 45 regions of our genetic code that could affect your severity. And these regions cause inflammation. They may affect your immune response. They may be associated with your blood group, and they might affect clotting. And some of these identified 3 possible therapies, of which one had collateral evidence from other studies that weren't genetic, that it might be a good treatment for severe COVID, and that was baricitinib. And then in our findings in the first study that we did suggested that baricitinib would also be good, and that plus the other evidence led to it being adopted in the recovery trial, where a year ago or so they published that it reduced mortality by 13%. So we learned from this that whole genomes give a more detailed, granular picture of the genetic code. They cover all 3.3 billion bases or letters that make your code. They cover all types of variation, and this bode well for other studies. So when we as a group of scientists and clinicians were getting together to plan the work packages of this consortium, it was considered that we should look at genetics too. So what have we done? Well, we've enrolled about 100 people with vaccine-associated clotting who also have low platelets and a specific immune response that you just heard about with platelet factor 4 antibody. We matched those to 500 people that were matched for age, their sex, and ethnically, not ethically, sorry about that typo, my apologies, matched controls. And all of them had to have an exposure to the Oxford AstraZeneca vaccine. So we collected clinical data and blood samples, and then we did whole genome sequencing, which is reading as much of your and my genetic code that we can do. But we also took samples so we could measure other biomarkers. For example, the genome is the code, but your RNA takes the message from the code and translates that into proteins. So we looked at the RNA, and then we looked at specific bits of the RNA in very great depth. And what this has allowed us to do in regions of your and my genetic code that affect your immune cells, we've been able to subset different groups of cells and look at different patterns. And then what we've done, as we've analyzed this, is we've been checking for similarities and differences between vaccine-induced clotting and people with severe COVID. Are there common factors between them? So what the questions that we're answering here are very important potentially for future vaccine development. So if this is not due to the COVID bit of the vaccine, but was due to the scaffold that holds the COVID bit, which is another virus called adenovirus, if it was due to the adenovirus, then that's vital for us to know, because it might affect how we use that virus to carry other vaccines in the future. So the analysis is now underway, and what I can tell you is our first analysis has found no common changes in the DNA that influence your susceptibility to vaccine-induced thrombotic thrombocytopenia, so vaccine-induced clotting. However, we are now looking at the rare variants, and because this has been said in the past to have a frequency of about 1 in 50 in the population, it's behaved 50,000, sorry, 1 in 50,000. It is behaving very much like a rare inherited disease. And so there may be rare changes in your and my DNA that we've yet to find, and that's what's underway at the moment. We have also begun to get a readout from some of the work on RNA. So I can say to you that we have found a particular signature that affects the immune cells. We, at the moment, are working out whether that's due to the vaccine and how recently you were vaccinated, or whether it could be a phenomena underlying the clotting that was experienced by patients in this situation. So we're still working through the data, and we hope very shortly, within the next 2 or 3 weeks, to have greater clarity on what the findings are that it may be influential. It could be that we'll find very little, but it is of interest that we have found some signatures that are in the immune cells that may be meaning that people are reacting differently, some people, to the presence of the vaccine or the virus that carries the vaccine into your cells. So it's still a bit of watch this space. This is really complex stuff to do, and some of the analyses we're doing have never been done with these approaches before like this. But we've got a great team and we're working well together. And I should take the opportunity also to thank everybody who's worked on this. What you're seeing here is first my deepest thanks to all of you who've been participants in the study or who have acted on behalf of a loved one or family member that you have helped enroll in this. The genomics work package comprises the people that you see on the left of the screen there, but I should also pay great tribute to Pip Nicolson, who's been very helpful in helping us to get some of the data and information together, and also in helping me manage access to the samples so we maximize their benefit for all of you. And I'd like to thank NIHR for funding us, and the MHRA, PHE, and of course NIHR for their support and advice, and of course all of you for listening. Thank you so much for having me here.
Well, thank you very much, Sir Mark. You've offered some tantalizing suggestion you might have some major insight in the next few weeks or months. So that's very exciting. And last but very much not least, we have Professor Paul Bennett with us. He's a clinical and academic psychologist who's worked in both the NHS and universities of Bristol, Cardiff and Swansea. He's published a lot of research about the psychological impact of thrombosis, and he's currently Emeritus Professor of Psychology at the University of Swansea. What he's known for among this community is his current work on looking at the psychological aspects of patients with VITT, and he's doing this with Thrombosis UK. Over to you, Paul. Thank you for coming.
No, my pleasure. Thank you very much, Beverley, and thank you for the consortium for inviting me, and for the people that took part in this study. I always forget this, so I want to make that in early, because I know quite a few of you are out there. I've seen names coming up in the various bits and bobs that have been circulating, so I really do want to say thank you very much to that. So without further ado, I will come to the paper that I'm going to present today. So the title of the talk for today is called Living with VITT, the Psychological Consequences, or Reporting the Blindingly Obvious. And for people that have gone through this, I think, you know, what I'm going to say today is maybe not new because you've lived it. And for many people, it's been a very difficult time. But I think in a time of disinformation and misinformation about VITT, I and Thrombosis UK were very keen that, you know, the reality of the experience of the consequences of VITT were published, got out there in a legitimate medical journal, so that we could actually provide some impetus into a lot of the talk that is going on about, you know, helping people with the consequences in terms of provision of services, financial support and so forth. So I think I personally have a very strong interest in this paper because I think it's really keen to get this information out. And in fact, the two studies that we're doing at the moment, oh, that's interesting. It's now not doing, we're doing one study, which is a qualitative study, which is talking to people that have experienced VITT themselves, and one study looking at people that are close relatives, loved ones of people who have had VITT. So these may be people where living with somebody where they are a survivor of VITT, or they may be sadly somebody that is living with somebody who unfortunately died from VITT, and we're very keen to get their stories out. And the first paper is now pretty much done. I'm hoping it will be published in the very near future, and it's the one I want to talk about, which is the survivors, the people with VITT, and the other is now in development as a consequence of that. So it's a very different type of study to what's been done before, where we've had thousands of people. This is 14 people took part in an interview where we talked about the experiences that they'd had as a consequence of having VITT. And what we did was we looked for commonalities in each of those experiences across that group, so that we could give a summation of the experiences that these people had. And what we did was to call them themes. So we identified two themes, really, one of which was their experiences at the acute period when they were diagnosed with VITT, or not diagnosed with VITT, as was often the case in the early days. And then what I've called surviving the long haul. And what I'd like to do is just to give some quotes from the people about each of these different themes, just to give a sense of what we're reporting in the paper. So the theme one was the trauma of care. One of the common experiences of the people that we interviewed were that they'd had multiple attempts to access and maintain care. And these are literally quotes from the people. They're nice quotes in the sense that they really reflect what people were saying, but they're obviously not nice for the people that went through. So the first was somebody's went to their GP and said, I think I've got VITT or vaccine-induced problems at least. And they were told, no, it's fake news. It's impossible. There's no chance you have. Honestly, it can't happen. And they had several attempts to talk to their GP before eventually going to A&E. There was a young woman who was diagnosed as having gas. She went to A&E and they said, you've got gas. And she was given oral antacid and asked to come back in 5 days. And she said, I was like, I've had a baby, I had a C-section, and after a C-section, you get tremendous gas. This was not gas. So he prescribed me a drug for gas that was obviously didn't do anything. I didn't have gas. And actually, this person went back to A&E and was still sent back home with antacids. And it wasn't until she went for a cervical smear that the nurse actually doing the smear, the practice nurse, said you really need to get to hospital, and eventually she got in. And there was this quote from somebody who was sent home from A&E, told she was okay, and then phoned up and said, your platelets are really low, so just be careful. If you cut yourself, you might bleed to death. Now, that's her quote. I'm not saying that's exactly what was said. But the issue here is that these people had a profound loss of faith in the system. They really did worry about whether there's you know, when they came, when they went into hospital, and whether if they went back into hospital subsequently, they will be treated and noted and identified as having had VITT. Now, most of these, to be fair, were in the early stages, but not all. Some of them were there when VITT was known and understood. As you can imagine, these people went into hospital with, you know, very frightening symptoms. They had a lot of anxiety in hospital. And here's a couple of quotes. I honestly thought I wasn't going to make it through the night. It was probably the scariest time ever because they wasn't explaining it to me properly. I'll skip bits. And every time I asked them, will I survive? You know, could this kill me? They said, we don't really know. We don't know enough about it, but you're on the correct treatment. And one person said, I knew they thought this would be the end of life because that was the only time he was allowed visitors. And he actually asked his wife to leave. He said, I'm asking you to leave because every time I just kept falling asleep and waking up, and every time I woke up, she was at the end of the bed and I wanted to go home because, to be honest, I didn't want her to be there when I died. So this is a time of tremendous fear and anxiety that, you know, lived on into the future in terms of post-traumatic symptoms and so forth. And what was worse for these people is that they were essentially doing this on their own. This was, don't forget, this is a time of COVID when things were very, you know, visiting and so forth was very problematic. And so both patients and partners experienced significant additional stress as a result of that. It was bad for me, but worse for my partner. And like, I woke up in the middle of the night, I remember crying, I just need a human touch. And I remember a nurse in ICU holding my hand and how much that helped me. So people were experiencing significant and profound anxiety. They had an illness that was barely understood, difficult to treat. They knew people were dying because some of these guys are going on Google and seeing some of the horror stories that were going on, and they were very emotionally unsupported by their friends and family. So this is setting up the sort of longer term issues that many of these people faced. Surviving the long haul, well, they were moving away from safety. So at one point, people were glad to be at home, but they were also very anxious. They, despite all the anxiety they had in hospital, they had become to have faith in it and to rely on it. And when they went home, it was, well, I'm not here. If there's any problems, what is going to happen? I can't get immediate medical care. Will I be all right? And as C noted, there was no point of call should her condition deteriorated, which would have been very useful. And what was also challenging for these people were that they would be going to medics or different people and often being treated with a degree of caution, shall we say, cynicism. Hematologists come out extremely well from this, you'll be glad to know, but a lot of other medics perhaps less well, again, maybe reflecting the early stages. But here was a neurologist who was sent a letter saying this patient has VITT, and she was told by him chronic migraines due to analgesic addiction, and you need to come off painkillers, and you have chronic fatigue syndrome. I literally walked out of that appointment crying. So people were struggling to get medics to understand, or other healthcare professionals. It was not easy for them. And of course, when they went home, some of these had profound physical problems. So, you know, here's somebody that struggling to walk 100 meters, was relying on family and so forth to cope. So there were a lot of challenges when these people went home. And of course, there was a continuity of symptoms that we all know about, you know, depending on where the clots were, where the bleeds were. There were a whole range of things. But here's just some, I think, that reflect some of the fatigue and the difficulties people were experiencing. And this is a nice quote that sort of reflects that. I can't concentrate like I used to. If I were to read a book, I can't remember what I've read. It's not I've got amnesia. It's just I can't concentrate long enough to take it on. You know, I used to be somebody who, like, if 10 people walked past me on the street, I could tell what they were wearing, whether they had glasses. And nowadays they could be running towards me naked and I may not know. You know, there's that brain fog, the fatigue, the difficulties that were common to just about everybody, regardless of the other symptoms that they were experiencing. And then there was that hope. You're waiting for the switch to go on and all of a sudden to feel better, and not knowing how you might feel your old self again. So people had that sort of ongoing symptoms that were incredibly challenging and very depressing, at the same time as there was that glimmer of hope that for many people may not have come along. And I think a dominant theme was the lack of support and wider recognition of VITT. I started off by saying we want this to go through a legitimate source as a reflection of these experiences. And, you know, people have been taken off Facebook, they've been taken off Twitter, they've been called anti-vax and so forth. And it's been very difficult for these people that we were talking to get their stories out there. Except, interestingly enough, there now does seem a whole slew of stories about VITT. So maybe this paper will add to those and get some more information out there. But I won't quote the moan about VDPS, etc. But the one on the right, some of the comments were hilarious. Yeah, they were. Hope you die. Somebody written, your own fault, it serves you right. I agree with that. The other one was repent. I got a few religious people. Yeah, you should repent. I can't. I don't think God wants to hear all that. Me repenting is quite funny, actually. And you get treated like an insane anti-vaxxer. So people trying to get their story out felt very aggrieved that this was problematic and difficult. And I suppose, you know, another overarching theme was this notion of multiple losses. And, you know, we all know the symptoms of those that can vary, but here are some of the sort of summations that people were coming up with. It sounds a little bit arrogant if I say it now, but there was a point where I genuinely was, and this in relation to where I am now, Superman. Whereas now I definitely feel like Clark Kent, who just, you know, he's clumsy, trips over things, he can't run. And an even more profound statement, I'm ashamed of myself. I'm ashamed I've become this. I was quite well known. I had a wonderful career. I missed that life because without that and with this illness, I can't see what use I am to anyone. And finally, I didn't feel invincible before, but I feel a lot less invincible now as a consequence of fear of problems coming again and the significant health losses, losses of jobs, losses of self-esteem that people are experiencing. Yeah, I won't go through that, so I shall leave it there because time's beating us. But I think this is what we're trying to get over to the readers of this. And I think what's been interesting is when I've given talks to medics, they said, wow, I hadn't realized the experience of these people. So, you know, I'm excited and looking forward to this being published.
Thank you, Paul. I think we all are interested in it being published. Pip, can I ask you to come back? And Paul, thank you very much. You're always such a wonderful speaker. Thank you.